Assalamu alaikum. Today we're going to be talking about DNA profiling. So let's begin. DNA profiling is a forensic technique used in criminal investigations in order to identify individuals by their DNA sample. The specific pattern that we obtain from the DNA sample is called the profile, and as it is unique, it helps us to identify the individuals. Interestingly, 99.9% of the human DNA is identical. However, there are certain distinguishing regions or distinct regions which we call the polymorphic regions or the VNTRs, the variable number tandem repeats. And these sequences are approximately 3 million base pairs in length. And in case of monozygotic or identical twins, the DNA is the same. Now let's have a look at the samples of three individuals. As you can see, the different individuals have different number of recurring VNTR units at the different sites. Say in individual number 1, we have 3 and 4 VNTR units at the different sites. And similarly, in individual number 2, we have 7 and 5 VNTR units in the different sites. And individual number 3 also has a different number of VNTR units. And basically, we call this variation polymorphism. You might be wondering who came up with the idea to identify individuals by their DNA sample. Well, it was geneticist Sir Alex Jeffreys who independently developed a DNA profiling technique in 1984. This technique involved microsatellites, also called as stutters, which are basically 10 to 16 base pairs of repetitive DNA. And the technique involved the simultaneous detection of these microsatellites to form a pattern, which we call the DNA fingerprint or DNA profile. Sir Alec used this technique to identify the culprit of a rape and murder case of two teenagers from the samples of 500 Sir Alec used this technique to identify the culprit of a rape and murder case of two teenagers from the samples of 500 local men. Before the actual process of DNA profiling, DNA must first be collected and then extracted in order to be analyzed. Collection may either be direct, for example through mouth swab, or it may be indirect, for example through the evidence or sample left by an offender at a crime scene. As for the extraction, the cell is bombarded with chemicals which cause it to break down, ultimately releasing the DNA, which is then isolated from the rest of the cellular components. There are many different techniques for the process of DNA profiling, and new technologies are emerging every day. The techniques that we'll be discussing in this video are the Rflp or restriction fragment length polymorphism, the STR analysis, and the amplified fragment length polymorphism or AMPFLP. The first technique that we're going to be discussing is the restriction fragment length polymorphism or RFLP technique. This begins with the extraction of the DNA from the sample, followed by the action of restriction enzymes which act as molecular scissors and cut the DNA into thousands of pieces of different lengths. Next, the DNA fragments undergo gel electrophoresis, where they are separated according to their size. For this purpose, they are passed through a porous gel, which acts as a sieve. And when electric current is passed through the system, the negatively charged DNA is pulled through the gel, such that the smallest pieces fall furthest away from the original position of the DNA. The reason for this is that the shorter fragments move faster and easier through the gel, whereas the longer fragments are difficult to move through the gel. Next, the DNA fragments are transferred to a nylon membrane, and then the single-stranded DNA is incubated with probes. Probes are basically fragments of mini satellites that have been tagged with radioactive phosphorus, and they bind to the complementary portion of the DNA, which in this case is the mini satellite portion of the DNA. Finally, the DNA fragments are visualized using X-ray technique, and we obtain the DNA fingerprint in the form of dark bands on a film. Let's have a look at the diagram for a better understanding. First of all, we'll extract the DNA, and then we'll use molecular scissors or restriction enzymes to obtain different lengths of the DNA fragments. Then we'll sort them according to their size and then transfer them to a nylon membrane, followed by incubation, and ultimately we'll obtain the DNA fingerprint. Now, to compare two DNA profiles, we'll run both the samples side by side on the same gel. The Rflp procedure isn't that reliable, so we've replaced it with other techniques. The reason for that is that we need a larger sample and the results take a lot longer, even up to a month. Resultant needs a very time consuming process and there is a higher chance of errors. Another technique that we're going to be discussing is the short tandem repeats or STR analysis, in which scientists usually examine 10 or more STRs, also called as microsatellites, which are basically 2 to 5 base pairs of repetitive DNA present at different loci. This technique also begins with the extraction of DNA from the sample, and this step is very sensitive, so even a small sample is sufficient, and it can come from a variety of sources including blood, saliva, hair, and even nails. No restriction enzymes are involved in this procedure. The next step is PCR, polymerase chain reaction, in which many copies of the desired DNA are made, and even partially degraded DNA can be copied in PCR. Next, the label primers come in. They bind to the complementary STR sequences, and because they have been labeled with specific fluorescent tags, they can be easily identified and recorded in the results. Now, once enough copies have been made, the DNA fragments undergo gel electrophoresis where they are sorted by their size. Followed by gel electrophoresis, they are incubated with probes, and finally they are visualized using a laser. The fragments are passed through a laser, and because of the specific fluorescent tags, we can see the output as a series of colored peaks, indicating the specific length and color of the different STR fragments. So let's have a look at the diagram for a better understanding. First of all, the DNA is extracted from the sample, then PCR replicates the fragments, labeled primers come in, then the fragments are separated according to their size, and ultimately we get a DNA profile similar to this. As for the accuracy of the method, if more STRs sequences are tested, then the results will be more accurate. Now, 1 in 10 million people may have the same STR profile, but as the world population is around 7,100 million, it is very unlikely for two people to have the same STR profile. If we have to identify a male preparator from mixed samples, we can do that with the help of YSTRs, which are derived solely from the male Y chromosome. YSTR analysis is less precise than autosomal analysis because the Y chromosome is almost identical in the patrilineal line. Otherwise, the YSTRs are processed the same as regular STRs. Another technique that we're going to be discussing is the amplified fragment length polymorphism or AMP-FLP. This technique involves shorter VNTRs than those in the Rflp procedure. They are approximately 8 to 16 base pairs in length. This technique also involves restriction enzymes to cut the DNA, and then the fragments are passed through PCR for selective amplification, followed by gel electrophoresis, which sorts them according to their size. The DNA fragments are then visualized using one of the following techniques: audio radiography, fluorescence, or automated capillary sequencing instruments. Now let's discuss the advantages and disadvantages of the procedure. The advantages are that it can be automated, it is not very expensive, it is highly sensitive, and the results are reproducible. As for the disadvantages, we require a high quality sample, and if there are any longer strands, they tend to bunch together so that they are harder to distinguish. As for the results, they are recorded as presence absence polymorphisms, and they look something like this. The procedure can help to identify the abundance, distribution, variation, and the role of the polymorphisms in the different species. Now let's have a look at the applications or the uses of the procedure. DNA profiling is important in solving crimes such as rape cases. It can help to link blood relatives and confirm their relations. It is also useful in paternity testing and to establish immigration eligibility. Furthermore, we can identify the victims of natural disasters and help them find their families. This method can also help to reveal the hereditary predispositions to certain diseases and birth defects, and it is also very popular in the research sector. Let's look at an example. Say we have to find the preparator in a rape or murder case. We can do that by taking DNA samples from the suspects and matching the samples with the evidence or the sample found at the crime scene. As you can see in this image, the dark bands of the crime suspect number one correspond exactly with the evidence sample. So we can conclude that crime suspect number one is the offender because his DNA profile matches the DNA profile found at the crime scene. Let's have a look at this next image, which shows that a child receives 50% of his DNA from his mother and the other half from his father. Moving on, we have the DNA profiles of a mother and a child, and we don't know who the father of the child is. We have to find out the father from three prospective dads. We'll employ the same technique that we used here, and as you can see, the bands from dad number three's profile have maximum correspondence with the child's DNA profile. So we can conclude that dad number three is the father of this child. The UK was the first country to set up a national database of DNA profiles, and this database contains the DNA profiles of the individuals linked to serious crimes only. And that's because in 2013, the DNA profiles of all the other citizens were deleted in accordance with the Protection of Freedom Act. There are certain controversies surrounding the process of DNA analysis, and that is because it focuses on the points of similarity only. And there are also certain rare cases where DNA may be contaminated or fake samples may be generated in order to frame someone else for a crime. For example, Dr. John raped one of his patients and then he implanted a drain in his arm which contained another man's blood. In this way, he managed to avoid the initial investigation, but ultimately he was identified as the culprit. Now, you may be wondering, what is the credibility of the procedure? Is it actually worth it? Well, let me tell you, the process has a very high success rate and false positives are very low. So, it's safe to say that DNA profiling is a very beneficial technique and it can help us in a variety of ways, as previously discussed. In this video, we covered the concept of DNA profiling, VNTRs, mini and microsatellites, as well as the history and techniques of the procedure, including the Rflp, STR analysis, and the amplified FLP. We also briefly discussed the applications of the procedure with examples, and then we discussed the storage, as well as the controversy and the credibility of the procedure. Re-watch the video to clear any concepts you may have missed, and thank you for watching.